Sexual reproduction involves fertilization of gametes. This is the fusion of the nucleus between the female sex cell and a male sex cell to produce a zygote, which is the fertilized egg. Some organisms can reproduce without fertilization, and this is known as asexual reproduction. This produces clones where the offspring are genetically identical to the parent because they have exactly the same DNA. To produce organisms that are genetically identical relies on mitosis, as covered in SB2. The advantage of sexual reproduction is that there is genetic variation in the offspring. This means that more offspring are likely to survive if the environment changes because some of those genetic variants are better adapted to survive in the new changed environment. The advantage of asexual reproduction is that it is faster than sexual reproduction as no mate is needed. Another advantage for asexual reproduction is that more offspring are likely to survive if the environment doesn't change. This is because if the parent is already well suited and adapted to survive and thrive in the environment, because the offspring are genetically identical, they will also be well suited and adapted to survive and thrive in the environment. The disadvantages for both forms of reproduction is the opposites of their advantages. For instance, the disadvantage of sexual reproduction is that it takes time to find a mate, so the reproduction rate will be slower. Another disadvantage is that fewer offspring will survive if the environment doesn't change. This is because there is genetic variation in the offspring, which means that some of those genetic variants will not be adapted to survive in the same environment as their parent. The disadvantage of asexual reproduction is that fewer offspring will survive if the environment changes. And this is because if the environment changes and affects one of the organisms, because the other organisms are genetically identical, they will also be affected. An example of these advantages for both forms of reproduction is shown here. An offspring from asexual reproduction are genetically identical to their parent, so if the parent plant is growing well, the offspring from asexual reproduction are also likely to grow well as they will be in the same condition as the parent plant. However, offspring from sexual reproduction will vary, and this means that some of those offsprings will not be suited to the same condition as their parent. In another example, seeds from sexual reproduction are spread by animals, so therefore those seeds will then grow in a different condition to their parent plant. Having variation means that some of these offsprings will be better adapted to grow in the different condition. So to summarize that, if the environment changes, such as a change in the temperature or a new pest or new disease, it is an advantage to have genetic variation because some of those genetic variants will be better suited to the new condition, and this increases the chances of survival. In asexual reproduction, it is faster than sexual reproduction because there is no need to find a mate, and this is a big advantage when there are lots of resources such as food available. An example of this is in aphids, where in the summer females reproduce rapidly by asexual reproduction where there is lots of food available. However, at the end of summer they reproduce sexually because those offsprings will then grow the following summer in a completely different condition, and because sexual reproduction is involved, there will be genetic variation, and some of those genetic variants will be better adapted to survive in the different condition. And here is a summary of advantages and disadvantages for both forms of reproduction with some more really good examples. Diploid cells are cells that have two copies of each chromosome, where one copy is inherited from the mother and the other copy is inherited from the father. Some cells are haploid, and this means that they contain one copy of each chromosome, such as gametes. Human gametes, the egg and sperm, contains 23 chromosomes. This means that they contain one copy of each chromosome, making the gamete cells haploid. When human gamete cells fuse together in fertilization, because they are haploid and contain one copy of each chromosome, the fusion in fertilization produces a zygote cell that contains two copies of each chromosome, making the zygote diploid. So the gametes fuse together in fertilization to produce a zygote that has got 46 chromosomes, or 23 pairs. And because it contains two copies of each chromosome, that zygote is diploid. And then during growth, when additional cells are added by mitosis, all other cells will also be diploid. And this means that they will contain 46 chromosomes, or 23 pairs in humans. So an exam question might be, explain why gametes must be haploid, and the answer is, gametes need to contain half the normal body cell number of chromosomes, so that when you fuse two gametes together, the zygote will be diploid and contain the same number of chromosomes as a normal body cell, which is 46 chromosomes in humans. Now we know that there are 23 pairs of chromosomes in a body cell in parents, which is a total of 46 chromosomes. This makes these cells diploid. If you were to fuse together a diploid cell inherited from both parents, you would end up with an offspring that contains 4 sets of chromosomes, which is a total of 92. This is genetically too many. We want an offspring that contains the same number of chromosomes as the parent cells. So they also have to be diploid in humans. The only way they could contain 2 sets of chromosomes is if one set was inherited from the mother and the other set was inherited from the father. So the problem is, how do diploid organisms produce haploid gametes that could be fused together to produce an offspring? Now we learned in SB2 that you could make cells using mitosis, but the problem with mitosis is that it produces diploid cells. So therefore you cannot make haploid gametes using mitosis. Instead, a different cell division is used called meiosis. And the stages of meiosis are not required for you to know in the exam. However, you need to know the difference between mitosis and meiosis. Meiosis starts off with a diploid cell, just like mitosis. And before the cell divides, there is also DNA replication. which we could recognize by the X-shaped structure of the chromosomes. And then the cell divides, and in the first cell division the chromosome pairs separate, so one chromosome 1 goes to one cell and the other chromosome 1 goes to the other cell. And that happens with all the other chromosome pairs as well. They separate to opposite cells. Where meiosis is different is that there is a second cell division, and this time the replicate chromosomes separate, which is similar to mitosis. And because there are two cell divisions, we end up with 4 genetically different haploid gametes. These cells are haploid, which means that they contain one copy of each chromosome. The DNA is also different, which means that these gametes are genetically different. So we can see in this animation, before the cell divides, there is DNA replication, as recognized by the X-shaped structure of the chromosome. And then the cell divides to form two cells where the chromosome pairs separate. And then there is another cell division. So this time 2 cells divide to form 4 cells, and this time round the replicate chromosomes separate, forming haploid cells. This means that we could start off with diploid cells and produce haploid cells using meiosis. And these haploid gametes can then be fused together in fertilization to produce our diploid zygote offspring. So in a 6-mark question where you have to compare and contrast mitosis and meiosis, you need to state the similarities and differences between both forms of cell division. Mitosis produces 2 daughter cells, whereas meiosis produces 4. In mitosis, the daughter cells are diploid, which means that they contain 2 copies of each chromosome, whereas meiosis produces haploid cells. This means that the cells have 1 copy of each chromosome. In mitosis, the daughter cells are genetically identical. By meiosis, the daughter cells that are produced are genetically different. To summarize that, we can say that mitosis produces 2 genetically identical diploid daughter cells, whereas meiosis produces 4 genetically different haploid daughter cells. So 2 cells are produced in mitosis, but 4 cells are produced in meiosis. The daughter cells are genetically identical in mitosis, but genetically different in meiosis. They're diploid cells that are produced in mitosis, but meiosis produces haploid gametes. Another difference is that there is 1 cell division in mitosis, so 1 cell divides into 2, but in meiosis the cell divides twice, from 1 cell to 2 and then 2 cell to 4. The similarities between mitosis and meiosis is that they are both forms of cell division. Another similarity is that before the cell divides, the DNA replicates. Now we know from SB2 that asexual reproduction relies on mitosis to produce the genetically identical diploid cells. However, we could then say that sexual reproduction relies on meiosis, and this is because meiosis is the only way to produce haploid gametes that could then be fused together in fertilization to produce diploid offsprings in humans. DNA is located in the nucleus of eukaryotic cells. It forms part of the chromosome structure that also contains proteins which holds the shape of the DNA together. The structure of DNA is a double helix that contains two strands. These two strands are held together by complementary base pairs. We can remember the rule for complementary base pair using the acronym GCAT, where guanine complementary base pairs with cytosine and adenine complementary base pairs with thymine. Other parts of DNA include the deoxyribose sugar and the phosphate group. The DNA molecule is made up of repeated structures called nucleotide, and one nucleotide contains a phosphate group, a sugar, and one base. A common mistake in the exam is to state that a nucleotide contains a complementary base pair, but it only contains one base. Now we know from SB1 that polymers are made up of repeating subunits called monomers. So because the nucleotide is a monomer, those repeating structures of subunits join together to make up the polymer DNA. And then the two strands are held together by complementary base pairs which have hydrogen bonds between them. And these hydrogen bonds are forces of attraction between opposite charges in the bases. Using the acronym GCAT, guanine complementary base pairs with cytosine, and they have 3 hydrogen bonds between them. And then adenine and thymine have 2 hydrogen bonds. You are expected to know how DNA can be extracted for your exam, even though it is not core practical. So first we have to understand that DNA is part of the chromosome, which is found inside the nucleus of a cell. The DNA extraction therefore requires detergent, salt, protease, and ethanol. The detergent is used to break down the nuclear membrane, and this releases the DNA from the nucleus. Salt makes the DNA stick together so that it makes it visible for us to see. The protease enzyme, we know, is an enzyme that breaks down proteins into amino acids. So therefore, we can use protease enzyme to break down the protein in the chromosome to release the DNA. And then we could add ethanol because DNA is insoluble in ethanol. And because it doesn't dissolve in the ethanol, it will then precipitate out. And the genome is all the DNA of an organism. And if we compare the genome of different organisms, we'll note that there are differences in the base. Gametes contain half an organism's genome because they are haploids. And because they're haploid, they only contain one copy of each chromosome, which means that they contain half an organism's genome. A gene is a DNA sequence that codes for a specific protein. This means that a gene contains the instruction to make a protein, and that instruction is in the DNA sequence. So if the exam question asks, describe the nature of genetic code found in the gene, you should understand that the genetic code is in the bases A, T, C and G. And that genetic code is the order of the bases. So in this example, the order is T, A, C, G, A. And that order of bases, the genetic code, gives the instruction of how to make the protein. And this leads on to protein synthesis. Protein synthesis involves two stages called transcription and translation. In transcription, a gene contains the genetic code, the instruction to make the protein. And that instruction is in the order of bases. So in this example, it's A, T, G, A, T, C, T, C, G, T, A. That order of bases tells us how to make the protein. And in transcription, you make a copy of the gene. And that copy of the gene is called mRNA. And because it's the copy of the gene, it will also contain the genetic code. So it will contain the order of bases. And then in transcription, we use that instruction, the order of bases, to put amino acids in the correct order. And we know from SB1 that if you join amino acids together, you can synthesize protein. So those correct order of amino acids are linked together to make a polypeptide chain, which will then fold into our protein. So to summarize that, the order of bases in the gene, which is the genetic code, the instruction to make the protein, is first copied in transcription to make mRNA. And because it's copied, that order of bases is now the same in mRNA, so it also contains the instruction to make the protein, and we can use that copy to put amino acids in the correct order. And that order of amino acid makes the polypeptide chain, which will fold into the protein. So the order of bases provides the instruction to put amino acids in the correct order to make the protein. So let's see how transcription works. First, the genes are located in the nucleus and the proteins are synthesized in the ribosomes. So if the instruction is in the nucleus and you make the proteins in the ribosome, that means that information to make the protein, the instruction, has to be sent to the ribosome. And this is why a copy of the instruction is made. The DNA is too large to leave the nucleus, so instead we make a copy of that instruction. And that copy is called mRNA. So first there is the coding strand, and that coding strand contains our gene, the order of bases that gives the instruction to make the protein. The opposite strand is called the template strand, and this contains complementary bases to the coding strand. To make a copy of the gene, you first have to break the hydrogen bonds to separate both strands. And then the template strand is used to add complementary RNA nucleotides. So using the acronym GCAT, we know that guanine is complementary to cytosine and adenine is complementary to thymine. So the next complementary RNA nucleotide to add is guanine, then cytosine. However, instead of a thymine RNA nucleotide being added, there are no thymine in RNA, instead it is replaced by uracil. then RNA nucleotides are continuously added one by one and joined together, and this makes the mRNA, which is the copy of the gene. And that mRNA contains the same order of bases as the DNA in the gene. So that means that it is a copy of the instruction to make the protein. And because DNA is too large to leave the nucleus, we can send the single-stranded mRNA copy, which has the same instruction to make the protein, to the ribosome. Now, transcription happens due to an enzyme called RNA polymerase, and it does four things. First, it attaches to a non-coding region in front of the gene. Then it separates the coding and template strand by breaking the hydrogen bonds. It then adds complementary RNA nucleotides, where uracil replaces thymine, and then these nucleotides are linked together to make mRNA, the copy of the gene, which now has the genetic instruction, the order of bases that can be used to make the protein. So to recap, RNA polymerase binds to non-coding DNA in front of a gene. It separates the coding and template strand by breaking hydrogen bonds. It adds complementary RNA nucleotides and then links RNA nucleotides to form mRNA. So we can see that here we've got the RNA polymerase, and it's bind to the non-coding region before the gene or in front of the gene, and then as it moves along the gene it separates the coding and template strand, and then using the template strand it adds RNA nucleotides that are complementary. Remembering that there is no thymine, instead in RNA is replaced by uracil. And then these RNA nucleotides are joined together to form mRNA. The mRNA then can be sent to the ribosome to make the protein. It has to travel out the nucleus and into the cytoplasm, and it does this through small holes in the nuclear membrane called nuclear pores. So recapping transcription, you first have your gene, and that gene contains the genetic code, the order of bases to make the protein, and then in transcription you are transcribing mRNA, which means that you're making a copy of the gene, and that copy is called mRNA. It contains the genetic code, the order of bases, and that mRNA leaves the nucleus through the nuclear pore and travels to the ribosome, where it provides the instruction to put amino acids in the correct order, which can be linked together to make the polypeptide chain that forms our protein. Now, for translation to work, we need to understand that proteins are made up of amino acids, and there are 20 different types of amino acids. If we compare these two different types of proteins here, we can see that they're made up of amino acids, but the order of the amino acids is different, and that's why the proteins are different. So in conclusion, different order of amino acids make different proteins. So if we put amino acids in a different order, the protein that is made will be different. So in translation, we've got our genetic code, the order of bases now in the mRNA, and when it binds to the ribosome, the ribosome moves along every 3 bases, and these 3 base triplets are called codons. And each codon codes for one specific amino acid. For instance, the AUG codon tells us that the first amino acid to add is the MET amino acid. So it's coding for the amino acid MET. The next codon, UGU, codes for a different amino acid, cis. So the cis amino acid is added next. And then the next amino codon, CCU, codes for the amino acid pro. And then amino acids are added one by one in the correct order based on the codon that codes for that specific amino acid. And then these amino acids are joined together to make your polypeptide, which will fold into the protein. So to summarize that, we've got our ribosome, which attaches to the mRNA, and the ribosome moves along every base triplet called mRNA codon, and then each codon codes for a specific amino acid. So AUG codes for the amino acid MET, so that would be the first amino acid in the polypeptide chain. Now that MET amino acid has to be transferred to the ribosome, and is transferred by a molecule called tRNA. The next mRNA codon is AAA, and it codes for the amino acid LIS. So that means that a tRNA molecule has to transfer the LIS amino acid next. Now there are lots of different tRNA molecules, so that means that different tRNA molecules or specific tRNAs will transfer these specific amino acids to the ribosome. And to make sure that the correct tRNA molecule is transferring the correct specific amino acid, these tRNA molecules not only contain the amino acid, but they also have a tRNA anticodon. And that anticodon is complementary to the mRNA's codon. So we can see here that the UUU tRNA anticodon is complementary to the AAA mRNA codon. And this is why this specific tRNA is transferring the life-specific amino acid to the ribosome instead of a different tRNA molecule, which could transfer a different amino acid. And we could see that from the previous example, the UAC tRNA anticodon is complementary to the AUG mRNA codon, and that is why the first amino acid that was transferred was MET, because the tRNA molecule with the UAC anticodon contains the MET amino acid. This means that specific tRNAs transfer specific amino acid to the ribosome in the correct order. And it does this based on its anticodon being complementary to the mRNA codon. So we can understand this using this example here. We've got our mRNA codon UGG, and that mRNA codon codes for the amino acid TRIP. And as we said earlier, specific tRNA transfers specific amino acids to the ribosome in the correct order based on its tRNA anticodon being complementary to the mRNA codon. So that means that one of these tRNA molecules has to transfer the correct amino acid, which we know is TRIP. So using the rule, guanine is complementary to cytosine and adenine is complementary to uracil in RNA, we know that the complementary bases are A, C, C. So that means that the only tRNA molecule that could transfer its amino acid is the one that contains an ACC anticodon, which is complementary to the UGG mRNA codon. And this tRNA will contain the trip amino acid, which is the correct amino acid coded by the UGG codon. As the ribosome moves along the base triplets, the codon, the transfer RNA or tRNA brings along the correct amino acids and put them in the correct order based on its anticodon. And these amino acids are then joined together to make the polypeptide chain. And this means that the mRNA codon has coded for a specific amino acid to join together to make the protein. And we can see that here. So we've got our ribosome, it attaches to the mRNA, and then that AUG codon codes for the MET amino acid. And the MET amino acid is transferred to the ribosome by a tRNA molecule, which contains an anticodon that is complementary to the mRNA's codon. And then the next tRNA molecule transfers the next specific amino acid because it's got an anticodon that is complementary to the mRNA's codon. And different specific tRNA transfers different specific amino acid based on its anticodon being complementary to the mRNA codon. And these amino acids are then joined together to make our polypeptide, which will fold into the protein. And we can see the same thing here happening in this animation, where the mRNA is binding to the ribosome and tRNA molecules are transferring the specific amino acids that are being coded for by the mRNA codon based on its anticodon being complementary to the mRNA codon. Now this is really important because if you got two different genes, they will contain different order of bases, and that means that they have different instructions to make the protein. And because they have different instructions to make a protein, that means that the amino acids are added in different order. And if different amino acids are added in different order, that means that when the polypeptide chain is made, it will fold differently into a different specific shape. And we know that shapes in protein are important. For instance, in an enzyme, the substrate binds onto the active site because they have the same specific shape. For example, the protease active site has the same specific shape as the protein substrate, and therefore only that substrate fits into the protease active site and can be broken down. The only reason why protease has this specific active site shape is because of the order of amino acids that fold in the polypeptide chain to make that specific shape. Lipase has got a different order of amino acids, so therefore it will form a different specific shape when the polypeptide chain folds. This means that the substrate that could fit into its active site will be different, and that's why only lipids could bind onto the active site and be catalyzed and broken down by lipase enzymes. So to summarize, there are lots of different genes in an organism, and these different genes are made up of different order of bases, that means that the instruction they have to make the protein is different, because the amino acids will be added in different orders. And so when those amino acids are added in different orders, they will make a polypeptide chain that will fold differently, and because they fold differently, they will produce different specific shapes in the protein, and those proteins will therefore have a different function. So therefore the order of amino acids will fold into a specific shape, and that means the proteins will have different functions, and that's why we know that those genes produce different proteins. So to summarize transcription and translation, transcription involves RNA polymerase binding to non-coding DNA in front of a gene. The polymerase then separates both strands by breaking the hydrogen bonds between the coding and template strand. It then adds complementary RNA nucleotides to the template strand, and then these RNA nucleotides are linked together to form the mRNA. The mRNA can then travel out of the nuclear pore into the cytoplasm, where in translation this mRNA attaches to a ribosome, and then complementary tRNA anticodons pair with each mRNA codon and bring specific amino acids which are added in the correct order, and those specific amino acids are joined together to form the polypeptide chain, and then the order of amino acids in the polypeptide chain determines how it folds into its specific shape, which determines the function of the protein. And using all that information, you should be able to answer all these questions. Now if we look at the UGU codon here, we notice that the middle base is guanine, and if there was a mutation in the DNA, that means when the mRNA is produced, there will be a different base, for instance UAU. Now this has an impact because now we've got a UAU codon, it will code for a tyrosine amino acid. So if it codes for different amino acids, that means that the order of amino acid has now changed, and because it's changed, the polypeptide chain will fold differently, and this will produce a different shape in the protein such that the protein will have a different function. So these changing bases are called mutations, and the mutations cause the proteins to have a different function if it occurs in the gene. Mutations could also have an effect if they happen in front of the gene. This is because we learnt in transcription that RNA polymerase attaches to the non-coding region in front of the gene. So if a mutation happens there, it will affect the binding of the RNA polymerase. Now the RNA polymerase function is to produce mRNA, which can then be used to produce protein because it contains the genetic code, the instructions to make the protein. So if there is a mutation in the non-coding region in front of the gene, it could cause the RNA polymerase to either bind more effectively, which will increase the quantity of mRNA produced, which will then increase the amount of protein that is produced, or it could affect the binding of the RNA polymerase negatively, and this will either stop the RNA polymerase from binding, which means that no mRNA and thus no protein will be produced, or it could decrease the amount of mRNA that is produced and therefore less protein is produced. So the amount of protein produced could be affected by a mutation happening in the non-coding region in front of the gene. Whereas, so an example of this is here using these questions. Question 7, men with prostate cancer may have an increased amount of a protein called PSA in the blood. What would be the explanation? or that would be a mutation in the non-coding region before the gene, and this causes the RNA polymerase to bind more effectively. So that means there's more transcription happening, more mRNA is produced, and therefore more polypeptide protein will be produced. However, if the mutation happens in the non-coding region before the gene, it doesn't necessarily make the binding of RNA polymerase more effective. It could also mean that the RNA polymerase doesn't bind as well. And if it does that, there'll be less transcription, less mRNA produced, and therefore less polypeptide or protein produced, as in the example for question 8. So in the biology specification, you need to understand how mutations have an effect in both the gene and in the non-coding region in front of the gene. So in specification 3.10b, we can see that if a mutation happens in the gene, it means that the order of bases is different, and that will code for a different order of amino acids, and therefore a different protein will be produced, affecting the phenotype. Whereas in 3.9b, if a mutation happens in the non-coding region in front of the gene, it affects the binding of the RNA polymerase. So if there is a more effective binding, more protein will be produced. If there is less effective binding, less protein will be produced. In specification 3.7b and 3.8b, you also have to understand how protein synthesis works in terms of both transcription and translation. So why do we have different eye color? This is because if you look at the eye color gene, you'll notice that there are some differences in the bases. So these change in bases are called mutations. And as we know from protein synthesis, if the mutation occurs in the gene, it could cause there to be a different order of base, and therefore there is a different order of amino acids, and that means that the protein that is produced will be different. However, if the mutation is in the non-coding region in front of the gene, it affects the binding of the RNA polymerase, so that either more protein is produced if the binding is more effective, or less protein is produced if the binding of RNA polymerase is less effective. Now, mutations or changes in bases cause some genes to have different versions. So both of these examples are the same gene, but they exist in different versions. So for instance, you've got a brown eye color gene and a blue eye color gene. So it's a different version of the eye color gene, and this is known as an allele, a different version of the same gene. Now we know that chromosomes come in pairs, because one copy would have been inherited from the mother and the other copy would have been inherited from the father. And because you inherit two copies, one each from both parents, that means that those two copies will contain the same gene. Now if you inherit a brown eye color gene from one parent and a brown eye color gene from another parent, that means that you can express brown eye color. However, what happens if you inherit brown eye color gene from one parent and blue eye color gene from another parent? Which color do you express, or is it a combination of both? So this part of genetics, we have to understand all of these key words, but more importantly the difference. So we need to know what a gene and an allele is, but the difference between them. We need to know the definition of genotype and phenotype, but also understand the difference. And then the same thing between homozygous and heterozygous alleles and dominant and recessive alleles. So gene is a segment of DNA or DNA sequence that codes for a characteristic. In other words, it tells you how to make a protein that has an effect on what is being expressed. An allele, as we said earlier, is a different version of the same gene. So for instance, you could have a blue eye allele and a brown eye allele. So it's the same gene, but a different version of it. A genotype is the genetic information that's in the gene. So in other words, the instruction to make the protein or the code. And then the phenotype is what is actually physically expressed using that information from the genotype. For instance, the eye color. Homozygous is when you have two alleles that are the same. So in the example earlier, if you were to inherit a brown eye allele from one parent and a brown eye allele from another parent, both of the alleles are the same. So that means that that allele is homozygous. Heterozygous is when both alleles are different. For instance, if you inherit a blue eye allele from one parent and a brown eye allele from one parent, you've got two different alleles for the same gene. So that means that the alleles are heterozygous. Dominant alleles are alleles that are always expressed, whereas recessive alleles are alleles that are only expressed if you have two copies of the recessive alleles. Now we could use all of that information to explain the phenotype and genotype of these three different people. So person 2 has the phenotype brown eyes because that is the color of their eyes that they are physically expressing. The eye color for person 1 is brown also, and then person 3 has got a different phenotype, blue eyes. And then if we look at the genotype, person 2's genotype is capital B lowercase b, and the reason why it's two letters is because there are two alleles, one of the allele inherited from one parent and another allele inherited from the other parent, and that's why we use two letters. Person 1's genotype is capital B, and person 3 is lowercase b. And then using the information we just covered, we should know that for person 2, their genotype is capital B, lowercase b. That means that both alleles are different to each other, and because they're different, this person is heterozygous for that allele. And the next person is capital B. Those alleles are the same, so we could say that this person is homozygous. And then person 3 has got lowercase b. Again, the two alleles are the same. Even though the alleles are different to person 1, because the two alleles are the same, they are also homozygous. Now if we look at person 2, they have got a capital B allele and a lowercase b allele. The capital B allele represents brown eye allele, and the lowercase b is the blue eye allele. And then if you look at their phenotype, they are expressing brown eyes. So that means that they contain a brown eye allele and a blue eye allele, and the brown eye allele is the one that's being expressed. This means that brown eye allele must be the dominant allele. So we can say that person 1, who has two capital B's, is dominant homozygous, because they contain two alleles that are the same, making them homozygous, and both of those alleles are the dominant alleles, so they are dominant homozygous. And we write those letters using capitals for dominant. Then if we look at person 3, they've got two lowercase b in their genotype. This means that they are recessive. So that person is recessive homozygous. And we know that for recessive to be expressed, you need two copies of the recessive alleles. And you can see that they are expressing blue eyes because they have got, that person has got two copies. Now we could use all of that information to then predict what genotypes our offspring will have. For instance, we first use a Punnett square, and in that Punnett square you write down the genotype of both parents. And the parents have got 2 alleles, so that means there should be 2 letters involved. And then in Punnett square you split those letters because only one of the allele is passed on to the offspring through the gametes. And that's because gametes are haploid, so they only contain 1 copy of each chromosome, so that means that only one of your chromosome set will be passed on to the offspring. And then you can look at the combinations to see the genotype of the offspring. So if we do an example, state the probability of having a blue-eyed offspring if you cross 2 heterozygous brown-eye people. So first we draw our Punnett square, and then one of the person is heterozygous, so that means that they're capital B lowercase b. They're heterozygous, so that means that they have 2 alleles that are different, and so we write the 2 alleles separately because only one of the allele can be passed on to the offspring. And the other individual is also heterozygous, so we do exactly the same thing. And then we look at the combinations they produce. So capital B, capital B, capital B, lowercase b, capital B, lowercase b. In Punnett squares, you always write the dominant allele first. And that's why it's capital B, lowercase b, and not lowercase b, capital B. And then the last possible offspring is lowercase b, lowercase b. And then looking at these combinations for the genotype, we can describe the phenotype, what is actually expressed physically. So here, this person is expressing brown eye because they've got 2 brown eye allele. This possible offspring will also express brown eye because they've got a brown eye allele and a blue eye allele, but the brown eye allele is dominant, so that is the one that will be expressed. And the same thing with this person here. And then if you look at the last possible offspring, they will express blue eye allele in the phenotype because there are 2 copies of the blue eye allele, and because blue eye allele is recessive, you need 2 copies to express it in the phenotype. And looking at the probability of having a blue-eyed offspring, it's 1 out of 4, so it's 25%. In the exam, they might ask you to write it as decimal place, so 0.25, or as a ratio, 1 to 3, or as a fraction, quarter. If they don't ask you what to write it, always choose percentage, which in this case is 25%. Now to do Punnett squares, you need to know additional information. So you may be asked about doing Punnett squares for diseases. So we're going to look at some example here. So this is Huntington's disease. And in Huntington's disease, if we look at the heterozygous, which is capital H, lowercase h, which is highlighted, we notice that an individual that is heterozygous will have Huntington's disease. And this means that the Huntington's allele must be dominant. And the reason why it's dominant because only one dominant faulty allele is required to express the disorder in the phenotype. You could have 2 copies of the faulty allele and express it, but if you have 1 copy, you will express that faulty allele because it is a dominant disease. The faulty allele is a dominant allele. Cystic fibrosis is different because if you look at the bottom, there are 2 lowercase f for someone who's got cystic fibrosis. That means that you need 2 copies of the cystic fibrosis allele to express it, and this means that cystic fibrosis is a recessive disease. So 2 recessive faulty alleles are required to express the disorder in the phenotype. So this is different to Huntington's disease where only 1 copy was required because it was a dominant disease. Here, 2 copies of the faulty alleles are required because it's a recessive disease. If you're heterozygous, that makes you a carrier. And in carrier, there is 1 faulty allele and 1 healthy allele. But the healthy allele is dominant, so that will be expressed. So carriers do not express the disease in the phenotype. However, they do contain a faulty allele which they can then pass on to their offspring. And using that information, you could then do Punnett squares, and you'll notice differences between a dominant disease and a recessive disease. Notice that you cannot have carriers in dominant disease, you can only have carriers in recessive disease where the individual is heterozygous. You can also do Punnett squares using blood group. So in this example here, if you're blood type A, you're expressing the blood type A antigen on the surface of red blood cells, and then someone who's blood type B will have type B antigens on their red blood cells. But if you're blood type AB, you express both type A antigens and type B antigens. So first we need to understand that a person who's blood type AB, they'll contain 2 chromosomes that are the same, and then because one of the chromosomes would have been inherited from the mother and the other chromosome would have been inherited from the father. And that means that there are 2 blood type alleles. So blood type A allele was inherited from one parent and blood type B allele was inherited from the other parent. So which allele would be expressed? And we know that the dominant allele is expressed, but in this case here, this person is blood type AB, so they're expressing both alleles. This means that both alleles must be dominant, and we call this codominance. So blood type alleles can either be IA, IB or IO, and everyone has 2 copies of the gene, so you only have 2 of those in a combination. IO is recessive to both IA and IB, and IA and IB are dominant, and if you're blood type AB, your genotype is IA-IB, and that's, you express both alleles, making you codominant. We should be then able to write down the genotype for all the other phenotypes. So if you're IA-IA, both alleles are the same, making you homozygous, and these alleles are dominant, so it's dominant homozygous. For blood group A, you could also be IA-IO, and that means that because both alleles are different, they're heterozygous. If you're blood group B, your genotype could be IB-IB, so again, 2 alleles are the same, so it's homozygous, and IB is a dominant allele, so it's dominant homozygous. We could also have IB-IO. Both alleles are different, so it's heterozygous. For blood group AB, the genotype is IA-IB, and because both alleles are different, it's heterozygous. However, both of these are dominant alleles, they're codominant, so we could say codominant heterozygous. And then the only genotype for those that have a blood group O phenotype is IA-IO. Both alleles are the same, so it's homozygous, and the IO allele is recessive, so it's recessive homozygous. And we could then use this information for Punnett squares involving blood group. For instance, if there are 2 individuals, person 1 has got blood group A and they're heterozygous, and person 2 is blood group B and they're heterozygous, that means their gametes will contain IA or IO for the person with blood group A, and the person with blood group B, their gametes will contain the genotype IB or IO. And when we look at the combinations, the possible offspring might have not IAB. Remember, there are only 3 different types of alleles for blood group, is either IA, IB, or IO. So that means that it will be IA, IB. There's no such thing as IAB. There must be two alleles for the offspring. And then we can look at other combinations. Remember to write the dominant allele first. And then if we look at the ratio, we'll notice that for IA-IB, this blood group is AB. And you got blood using the rule, you got blood group A, B, and O. So it's a 1 to 1 to 1 to 1 ratio, or 25% chance of each blood group. And you should be then able to do Punnett squares for blood group using those information. In the exam, they could ask you to explain things in addition to do Punnett squares. So ABO blood groups inherited using codominance and multiple alleles. Explain how ABO blood group alleles illustrate dominance, recessiveness, and codominance. Where it illustrates dominance because the A allele and the B allele are dominant over the O allele. And that means that the O allele is recessive. It illustrates codominance because the A allele and the B allele are both expressed in the phenotype if they're together. And that means that they are codominant. Which parent determines the sex of the offspring? A male has sex chromosome XY and females have the sex chromosome XX. If you do a Punnett squares for these sex chromosomes, you'll notice that 50% of them are XX and 50% of them are XY. And that's why 50% of the population is male and 50% roughly is female. Now if you look at combinations, if the father's sperm contained X, the mother's egg only contains X, and therefore it can't change the sex of the offspring. If the father's sperm contained Y, again the mother's egg contains X sex chromosomes, and so it can't change. It could only end up producing offspring that are XY in terms of their sex chromosomes. If the mother gives an X chromosome in its egg, the father's sperm could either contain an X chromosome or it could contain a Y chromosome. So therefore, it can have an effect on whether the offspring is male or female. So in this question, you should recognize that humans contain two sex chromosomes that determine the sex of the offspring. Males have XY sex chromosome, where females have XX sex chromosomes. Only the sperm cells contains a Y sex chromosome, and therefore males determine the sex of the offspring because it's male if the zygote gets a Y at fertilization, but female if the zygote gets X at fertilization. The eggs only have X chromosome, so therefore it can't change the sex of the offspring. Now as we know, chromosomes come in pairs because one copy is inherited from the mother and the other copy is inherited from the father, and a female has two X chromosomes. This means that it contains two copies of every gene on that X chromosome. If one of the X chromosomes inherited contained a faulty allele which expresses a genetic disorder, it will contain another X chromosome which will be expressed instead if that was the dominant allele. However, in males it's different because male sex chromosomes are XY, and the Y chromosome has a section missing. This means that there is only one allele for that specific gene in the missing section. If there's a faulty allele inherited in the X chromosome in males, there is no second allele in the Y chromosome for those genes. So chromosomes come in chromosome in diploid cells come in pairs, and that means you contain 2 copies of genes. However, in the Y chromosome, those genes are missing. So for male, they will only have 1 allele for the gene because that gene is missing in the Y chromosome. If that allele is faulty and causes a genetic disorder, that means that the male will develop the disorder or the disease. Females have 2 X chromosomes, so if they inherit a disorder allele, they might have a healthy allele that is expressed instead if the healthy allele is dominant. If they inherit 2 recessive disorder alleles, then they will develop the disorder. However, the probability of inheriting 2 disorder alleles is less than the probability of inheriting 1 in males. So that means that there is a higher probability of males developing a disorder in their sex chromosome, and this is known as sex-linked genetic disorders. So to summarize that, sex-linked genetic disorders are more common in male because they only have 1 X chromosome. Females need to have the gene on both X chromosomes to develop the disorder. If the female is heterozygous, that means that they are carrier, so they will appear normal, but they can pass on the faulty allele to their offspring. An example of a sex-linked genetic disorder is color blindness caused by cone receptor cells that don't work properly in the retina. So if you had to do Punnett squares, you should be able to analyze them. So you can see here, here's the mother, and the mother's genotype is X capital R, X lowercase r. So because the mother has 2 X chromosomes, we could say that the mother is female. And we could also say that because the alleles are capital R, lowercase r, that means that they are a carrier. The father's sex chromosomes is XRY. So because the father is XY, the father is male. And there is no allele in the Y chromosome because that part of the gene or section of the chromosome is missing. So this father here has only got 1 allele, which is the capital R, which is the healthy allele. And then we can look at the possible outcomes of the offspring. So the first offspring combination is female because it's XX. Next one's male, then female, then male. And then if we look at the allele, we've got 2 capital R's, so that's healthy. The next possible offspring is just a capital R on its own because there's only 1 allele. There is no allele on the Y chromosome, so capital R is also healthy. The next person is capital R lowercase r. That means that they are carrier because they have 1 healthy allele and 1 color blindness or red-green color blindness allele. And then the male is just lowercase r on its own. Again, there is no second allele because the Y chromosome doesn't have that allele. It's missing in its section of the chromosome. So therefore, because it's only got lowercase r, it will have to express and develop that disorder. So this male has got color blindness. In exam, if you're doing Punnett squares for sex-linked genetic disorders, always write the X and Y first and then write the letters for the disorder afterwards so that there's no confusion. Notice that males cannot be carriers for these sex-linked genetic disorders. They either express the disorder or they don't, and they're healthy. Only females can be carriers. So an exam question, explain the difference in inheritance of haemophilia by males and females. We know that males inherit their Y chromosome from the father, So that means that if they're inheriting hemophilia, which is a sex-linked genetic disorder, they will inherit that allele from the X chromosome from their mother. And they only need one copy of the sex-linked genetic disorder allele to express it, because there is no second allele in the Y chromosome. For females to express these sex-linked genetic disorders, they need two copies. So that means they would have to inherit those two alleles from both their parents. So this means that males only have one allele for the gene and females have two. So females can only develop the disorder if they inherit those two alleles from both parents, where males only need to express the disorder if they inherit one copy of it from their mother. Now, other than Punnett squares, you also need to know how to interpret family pedigree charts, and this involves the use of squares for male and circles for female. Always use the key to understand what or how the disease is represented. And looking at these questions, which letter shows the recessive allele, which we know is written in lowercase, so here it will be Q. Why doesn't Caroline have PKU? So if you look at Caroline's genotype, is capital Q, lowercase q. So capital Q is dominant and it's the healthy allele. So that means that the dominant healthy allele is expressed. For Brian's genotype, if you're using the key, we know Brian is a male with the PKU disorder. So to express PKU, it's a recessive disease. That means that Brian should have two lowercase q's. And then the next question, calculate the probability that Sam has PKU. So if we look at Sam, Sam is the son of Brian and Caroline. We know that Brian is lowercase q, and Caroline is capital Q, lowercase q. So if we put that into a Punnett square, the probability that Sam has PKU, which is the genotype lowercase q, there are only two possibilities out of 4. So that means that is 50% chance. A long time ago, scientists believed that variation in parents blended or fused together to produce the offspring. For instance, if there was a plant with a white flower and another plant with a purple flower, the offspring would produce pink flowers. A famous person called Mendel did experiments on inheritance to discover rules, and what he did is he crossed pea plants of different height, so a tall pea plant with a dwarf pea plant, and all the offsprings that were produced were also tall. And then he took 2 of these tall pea plants from the offspring and crossed them together, and he was expecting the offspring to also be tall, but he found that some of them were dwarf plants. So suddenly there was a variation that appeared which can't be explained by blending. So Mendel showed that characteristics don't blend. Now we know, using the ideas of genetics now, that he must have crossed a homozygous tall plant, capital T, with a homozygous dwarf plant, lowercase t, and that produced offspring that were heterozygous, capital T, lowercase t. And then these heterozygous were crossed together and they produced offspring, and a quarter of those offspring were lowercase t, producing dwarf plants. So in this specification, you have to describe the work of Mendel in discovering the basis of genetics and recognize the difficulties of understanding inheritance before the mechanism was discovered. So all you need to understand is he crossed a homozygous tall pea plant with a homozygous dwarf pea plant, and that produced tall pea plants as well, and then 2 of these offsprings were crossed together and he managed to find some dwarf pea plants in the offspring. So he summarized his work forming 3 laws. Each gamete receives 1 factor for characteristic, Well, we know that one factor is now called a gene. In other words, the gamete will only contain one gene of a characteristic. So if we got a diploid cell, only one of the allele will move into the diploid, because we know in meiosis, after cell division, those gamete cells are haploid, so they could only contain one copy of the gene. So that means it will either contain a capital T or lowercase t using the example. Then the version of a factor that a gamete receives is random and does not depend on other factors. So that just means that one gene has no effect on another gene that's inherited. So it could be that having a tall allele means that what's being inherited will not be, say, a different characteristic in the P's. And then his third law, where some versions of a gene are more powerful than others and always have an effect in the offspring, we know that different versions of a gene is called an allele. And if some of these alleles are more powerful, that means that they must be dominant. Now, using Mendel's idea, we could see that inheritance is more understandable now. Now, most phenotypes are results of multiple genes rather than a single gene inheritance. An example is skin color. We could see that it's caused by 3 different genes, and this makes it complicated if you're doing a Punnett square, which you're not expected to do at GCSE. All you have to understand that the phenotype for most characteristic is usually due to several genes and not a single gene. If there was a change in the base, we know that that's called a mutation. If you look at the CCA codon, it codes for the amino acid Pro. If that adenine base changed to a guanine, it will also code for the amino acid Pro. So that means that there was no change in the coded amino acid, so that the order of amino acid is still the same, and this means that the same protein is produced. So here's an example where most genetic mutations actually have no effect on the phenotype. However, some mutations have a small effect, and then very rarely does a single mutation have a very big significant effect in the phenotype. In 2003, the first complete human genome was decoded, so that means that they sequenced all the bases in a human. A genome is all the DNA of an organism, so we know that the genome in a human is different to, for example, the genome of a banana, because they contain different DNA sequences. Now, the use of knowing the DNA sequence is that first we could find out if there's a risk of developing a disease, and then we could also identify which medicines can be used to treat a person's illness. So in this example here, we could see whether a person is at a risk of a disease because we could find out if they've got a mutation that causes the disease. And then we could also see whether the medicine will work or have bad side effects based on the alleles that they contained after doing DNA sequence. Now, siblings inherit DNA from the same parents, so why do they look different if they're inheriting DNA from the same individuals? Now, we know in meiosis the cell divides twice, and here, for instance, if a person had a brown eye allele and a blue eye allele, after cell division they will form gametes, and because these gametes are haploid, they'll either contain the brown eye allele or the blue eye allele. So that means that when one of those gametes produce the offspring, you could produce some offsprings that have got brown eyes and some offsprings that have got blue eyes. And that is because different alleles can be inherited during sexual reproduction, because meiosis produces gametes that are genetically different. Now, identical twins are genetically identical, so why do they look different? This is because of the environment. So characteristics that show variation caused by the environment, if that change happens by the environment during the lifetime of the individual, we call it acquired variation because they acquired it during their lifetime. An example would be having a scar. So that is an environmental variation. Some people have a scar, some people don't have a scar. Having the scar was acquired during the lifetime, so it's an acquired variation. You should be able to tell if a characteristic has got genetic variation or environmental variation. There is genetic variation if the genes are not the same, and there is environmental variation if the environment is different and has an influence on what characteristic they have. For example, hair length is an example of an environmental variation because the length of hair depends on how much hair has been cut off. Blood group shows genetic variation because the blood group is based on the genes that are inherited from a person. So if we inherit different genes, that means that we will express different blood group. So that means that there is variation genetically. And then some characteristics are both. For instance, height shows both genetic variation and environmental variation. It shows genetic variation because the height that you could grow to might be based on the genes that you inherit. And then to reach that height will be based on the environment in terms of how much good nutrition you get to grow to a certain height. So in an exam question, describe the causes of variation that influence phenotype. You could say that genetic variation causes different characteristics due to either mutation, where there's a change in the base, or due to sexual reproduction, where different alleles can be inherited. And then the other type of variation is environmental variation, where different characteristics are caused by an organism's environment. An example might be acquired characteristics during a lifetime of a person.